We'll begin this discussion by giving you a definition and talking about the ramifications of what a protein domain is. Hopefully, you're somewhat already familiar with this concept, with this vocabulary word. But basically, a domain is a definable region of a protein that has a very specific structure and a specific function. And we can draw an analogy with a domain to the morphology of organisms. So really, with domains, we're looking at the molecular level. But for instance, in vertebrates, we have these recognizable components of vertebrates that can be compared. So we can compare the forelimb of a fish to the forelimb of a chicken to the forelimb of a lizard and a human and so forth. And that is a subcomponent, a subunit of the overall physical manifestation of that organism that we can connect together, and it's very useful to help us study them. And so in the same way that a arm or a forelimb more generally is a subunit, a component of a vertebrate, a domain is a recognizable subunit of a protein. And they are recognizable across many different types of proteins. And in fact, domains are how we identify connections between proteins and how we define protein families. So protein family is largely defined by the types of domains it has. So here I've got a diagram, and it's very diagrammatic. The line represents the total protein. The different shapes and colors represent regions that are different protein domains. The shapes are just kind of randomly chosen to stand out from the other shapes. But notice that in these four very different proteins, they all have this SH2 motif. And not only that, but it is a duplicated, right, a dyad of this SH2 motif, even though there's quite a bit of space between the dyad in the P85 alpha. So this unites them and shows that that component at least, maybe other components, but this component of the protein shares a common evolutionary history. Okay, they are connected to one another. And often there is incredible amounts of conservation to the amino acid sequence within these protein domains. And they have a conserved function. Sometimes there's, in some protein domains, there's very, very constrained function where every time you find it, it's pretty much doing the exact same thing. In other times, there's a little bit more of a relaxed but still constrained function, so that there might be a range of chemical functions that that domain has, but they're all kind of within the same general area. So we might see that domain serving as a binding site for one type of molecules in a certain protein, and then it serves as a binding site for a related type of molecule in another protein. So still same basic job, but with maybe some slight modifications. So we have some constraint and some flexibility, and the different domains really are going to show different amounts of that flexibility. Now, within protein domains, we also have motifs. And a motif can be defined both based on its amino acid structure, and it can be based on the amino acid sequence is what I meant to say, and it can be based on the physical three-dimensional structure of that amino acid sequence, right, of these polypeptides, which is just a fancy term for a protein sequence. Okay, so polypeptides make very specific, unique structures. In this case, this is an alpha helix turn alpha helix. And so we get these very clear repeated structures. And they often have, like I say, large amounts of sequence conservation. Okay. Now, sometimes a motif corresponds on a one-to-one ratio with the domain, so that entire thing can be a domain, but more often we can break entire domains. Let's say we've got a enzymatic binding site, or we have a transmembrane domain, right? The part of the protein that helps to embed it solidly into the membrane of a cell. And within that transmembrane domain, we might see very similar repeated motifs. Many transmembrane domains are made up of 4 or 5 or more repeated repeated motifs that all then bind together and add up to the final domain, which creates that transmembrane portion of the protein. Okay, so motifs can be and mostly are smaller than the overall domain, But in some cases, for small domains, they may only be the motif and the domain may be roughly equivalent. Now, these protein domains usually, although not always, but they usually fold independently. Now, what does that mean? During the gene expression process, mRNA is translated into an amino acid sequence, and we get this polypeptide bond that forms one amino acid after the other. And as soon as that is complete, even sometimes before it's complete, that primary structure of the protein, right, the sequence of the amino acids, begins to naturally fold over and form the secondary and then the tertiary structure. And then, of course, if there is a more complex interaction of the polypeptide strand with other polypeptide strands, we can get quaternary structure of proteins also. So that's something you should familiarize yourself with if you're not. Review what is meant by primary, secondary, tertiary, and quaternary structure of a protein. And by when we say that they fold independently, what we mean is if you put them in physiological conditions, the right range of temperatures, the right range of pH, so on, all of these things, salts that make up the environment in which they are being used, they will fold into their correct shape nearly always on their own. And they do this based on the chemical interactions of the different molecules, the different atoms in each amino acid, and how they interact with one another through hydrogen bonding, through van der Waals, all those great chemical forces that can cause different atoms and molecules to interact are set up and have been selected for over billions of years of evolution to fold into their appropriate and functional shape. And of course, with proteins, shape is function. They have to be the correct shape to be able to do their job. Okay? So, generally, we can treat these domains as independent elements. Although, as we'll see when we review in the next lecture, when we review the Bagowski paper, there are sometimes some coevolutionary things that we need to consider where domains may have an effect on one another. Two domains from different proteins might have an impact on each other's evolution. But basically, we treat them as independent elements. So in a very real sense, the study of protein domains is a reductionist approach to the study of the overall evolution of proteins. So that's a good vocabulary word to learn. A reductionist approach is essentially taking a large complex problem, breaking it down into subunits that can be handled independently, trying to then understand each of those subunits, and then the overall goal is that better understanding of those subunits will then be able to connect them back together and be able to synthesize what's going on in the whole. So by understanding individual domains and the way that they evolve, the way that those changes then impact their own structure and function, and then how that change in structure and function impacts and affects the rest of the protein and maybe even domains in other proteins. So we begin to synthesize and put it all back together to get a complete understanding. So notice we have two very similar proteins here. This is from our original example that have the same basic structure at the beginning, these two SH2 domains, right, the duplicated version. And then they have different enzymatic activities that are tied to those. And so we can assume that this portion of the protein is probably going to have a very or maybe even completely conserved function. But then we get variety and diversity of function in these others. And it would be interesting then to study these PTPase and kinase to see if they perhaps share an ancient connection and then have evolved to take on different function, while the rest of the protein has remained pretty much constrained and still maintains the old ancestral function. And so just like we can get parts of living organisms that remain relatively unchanged for millions of years, billions of years, and then other parts of them evolve, the same thing can happen with domains. Some domains may be very conserved and locked into a very narrow range of function, and then other domains on the same protein are able to evolve and change more flexibly. And so this component nature of proteins allows for flexibility among rates of evolution. And of course, those flexibility and rates of evolution are going to be primarily down to natural selection and what has conferred an advantage among those different domains in the past. And so we can define this these independent elements both genetically at the DNA level, but usually more easily at the amino acid level. And then we can also define them functionally, meaning do they maintain their function? And there is a connection and a relationship between genetic conservation and functional conservation. Now, obviously, if we maintain genetic sequence and have very little change, then function is not going to change, right? Because function is dependent on sequence. But the reverse is not necessarily true. Sometimes we can quite drastically and significantly change the genetic makeup, certainly at the DNA level, sometimes even at the amino acid level, but still maintain functionality. So there are some instances where there's a lot of flexibility, and it's kind of this idea that for some chemical processes, there's really only one way to accomplish whatever outcome we are examining. And so then we would expect to see a large amount of constraint in the molecules that are involved in that chemical process. However, in other cases, there might be 4, 5, or 6 different effective ways to accomplish the same task. And so we might see a lot more diversity in the genetic structure of those more flexible parts of proteins, but they still maintain or largely maintain the same overall function. So that tells us a lot about both the evolution and the nature of that chemical reaction that is being mediated by the protein. Now, we define, as I said earlier, proteins by their domains. And some protein families are incredibly large. So here we have the arid family that is defined by this 1140, I believe it is, a little more, a little less, base pair. Let's see, I'm sorry, 140 base pair red motif. And we see it over and over again in all of these. Now notice that there are subcategories or subfamilies. So here we have part of the arid family, arid 4A and 4B, that are further defined by other motifs that they have in common. Same thing here. This is why they are given numbers and then letters. So we have both large families defined overall by classes of classes of domains. And then within that, we can define subfamilies based on even more shared similar domain patterns. Now, one of the very interesting things is we are now able to examine pretty much, well, not all, but a large chunk of the protein universe. And in some species, we pretty much have been able to study to a fairly significant degree the entire protein complement within that entire species. And what we have found is that there are some domains that are incredibly common that we see over and over and over again and define these really big families. In fact, we are even calling some of them super families because there are just so many proteins that contain those domains. And then there are other proteins, domains, that are maybe only a very narrow range. We might only see a handful or maybe even only 1 or 2 genes. They may even be confined to a smaller portion of the overall biological diversity. So we can define the success and size of a protein family both in how many members of that family are found in a single species and how widely distributed that family is across the diversity of all life on Earth. And because all life on Earth shares a common ancestry, we find what we would expect to find, that there are some domains that are conserved across all life on Earth. You can find them in bacteria and archaea and all types of eukaryotes. There are some domains that are specific to eukaryotes. And there are some that are found across all life, but then kind of become massive and diverse in one group and remain relatively small and not as important in another group. And so this tells us a lot at the molecular level about the evolution of these different groups and their protein complements. Now, there are many resources. We are not going to get into the mechanics practically of how these are done, but there are lots of resources. These are 4 of the biggest ones that allow you to investigate and look for matching protein domains, see what part of what family a gene may belong to, see if there are similar domains or maybe even similar groups of domains in other proteins and other species. And so we have a ton of resources that have been developed, and they're getting better and better as our databases become more and more complete. And so we are both increasing the number of species that are represented in this database and increasing the a number of genomes that we've categorized fully, or at least pretty much fully, where we know and have identified every single protein in those genomes. So these databases become better and better at defining those interconnections, which really, as we talked about in the previous lecture, is really an exercise in homology. But now we are establishing homology of subunits of proteins. And because of this, you can see some really interesting patterns. And I'm going to show you here in this next one. So here's our homology, and the red region shows a DNA binding motif, right? This is a motif that leads to this functional unit in this gene of binding to a specific sequence of DNA. And so notice that it's really, really conserved, especially the ones that are in red. I think the ones in red are 100% conserved. Every species in this sample, or maybe not 100%, but it looks like at least maybe 80% are completely conserved. And then there are a few standouts. But notice we see lots and lots of red here. And although there is similarity in the rest of the gene, particularly at the beginning, this level of similarity is much, much lower, and there's much more flexibility in how this part of the gene has evolved, as opposed to this part that is very, very constrained to perform a very specific function. Now, when we see this level of constraint on a domain, it means one of two things. First off, it means that this is a very, very important function of this protein, maybe even central to what that protein does. And so if we change too much about it, it is no longer able to serve its core function. So this is almost certainly central to what this protein does. In this case, these are transcription factors that need to bind to target DNA to influence the expression of target genes. And so if we cannot bind to DNA, then we are no longer a transcription factor. And so that has maintained the conservation of this region of the gene and then allowed others to evolve. Now, the second thing, and these can both be together, but the second thing that this might mean is that perhaps this has a very narrow range of functional, well, let's put it this way. There's a very narrow range of the way to get that job done, that maybe that's the only specific way to do it, because any changes, or at least most changes that have come into here, are not kept. So not only is this an important function of the gene, but if we see this huge level of conservation, it probably means that there's a very narrow range of diversity and variety that will allow that domain to accomplish its main purpose. And so it becomes more difficult to evaluate domains that maybe have a more promiscuous, meaning that there are more ways to accomplish the same end outcome. And there are some domains that are like that. And so we look at them at the genetic level, and they're quite diverse. But then when we look at them at the functional level, they're very, very similar. And so some genes, or some domains, we can recognize very, very easily at the amino acid level, like this DNA binding motif here in these Hox genes. Others are not as recognizable at the amino acid level, but once we look at how they fold and what function, maybe even assess empirically the function that they have, we can recognize and find those connections. So it really sometimes takes a lot of study and characterization to completely identify all of the domains that are present in an organism or in a group of organisms. And so domains can diversify. And now we are attempting and figuring out and understanding the regions of the domain that are very, very constrained. We can do that again, both based on sequence and on function. So here they've got kind of this cloud of the overall protein. And basically what this is an overlap of where we would expect the different amino acids to sit across many, many different homologous, orthologous versions of this protein. And notice some of them pretty much always fall in the same place, and so it looks very kind of defined. In other parts, it's much more diffuse, and so proteins might have a wide range of where they're going to sit, and then different color changes may even have different proteins that could be in that area. So we can think about it in terms of different amino acids in that area. We can think about it in the terms of amino acid and amino acid substitutions, but then we can also think about it as how that amino acid physically changes the three-dimensional conformation of that protein. And so there are multiple ways to do this. And again, as our databases get better and better, we can do that. But we can identify parts of this protein that are very obviously critical to its central function and have been conserved and maintained over millions of years of evolution, and other parts that have been evolving much, much more frequently. Now, one last thing I want to say about this area that is diffuse. Just because it's not completely conserved doesn't mean it doesn't have an important function. But what it means is whatever important functions it may have, that function is probably going to be different from one protein to another. So for instance, in this example back here, if we were to do the same sort of thing, this is a very simple diagram for these SH2 containing members of this family, but we would probably expect this part of the protein, whatever three-dimensional shape it folds into, to be very, very conserved, and then these other parts to be much less so, and maybe even be very diffuse, so we can't really see much physical similarity in the way that they fold at all. But that doesn't mean that these are not important functions. It just means that the functional work of this part of the gene has diverged, where the functional part of the rest of the gene has remained very, very conserved over time. Now, you notice this with the SH2, that often domains duplicate. This is one of the most common ways for genes to evolve, is to duplicate small functional components of them. And in fact, this is another thing that I talk about in my undergraduate course. We haven't pointed this out here. I guess maybe I'm taking it for granted, but let's go through it. In many cases, when we draw comparisons between these protein domains and the eukaryotic genome, what we find is that the exons often correspond fairly significantly to protein domains. So not only are these domains functional, identifiable components of the final protein product, but they are also these spliced in components of the overall eukaryotic gene. And the reason for that is there has been positive selection pressure for increasing this association between domains and exons because of this domain duplication and domain shuffling process. So I always tell students it's kind of like cannibalizing or using old machines for parts. So if you had a whole bunch of old cars, for instance, it's possible that if you took functional components out of those cars, you could put together a working whole. So you pull the engine out of one car and the suspension out of a second car, and maybe the transmission out of a third car. And you put them all together, and you might have to do a little bit of shifting and modification, but you have a much better chance of having a functional product than if you were to take, just say, some sort of laser cutter and lop off a random section of one car, weld it to a random section of another car. You're almost certainly going to end up with junk if you do that. And so this association between domains and the introns and exons increases the chances that when a mutation shuffles parts of a gene, it's going to shuffle functional parts, which, again, the most of the time, random shuffling even of functional parts is not going to give you a positive outcome. But there's a much, much higher, albeit still a fairly minuscule chance of having a functional outcome if we have exons associated with the functional domains. And so what this means, and we'll talk about processes in the next discussion, but there are processes that can increase the number of domains in a protein. So if we have two SH2 domains, or in this case 5 domains as designated by this triangle, these did not all arise independently to end up with the same structure, right? They arose originally as a single one, maybe did an okay job, and then it was mutated, so we had a duplication of just that region. And getting an extra copy of that resulted, in this case, just by chance, in it doing its job more effectively and more efficiently. And so domain duplication is a very important and critical part of gene evolution. And so just like duplication of genes can impact the functional properties of the organism and the patterns that we see in the genome, the duplication of domains can impact the functional properties of each gene, and then, of course, increase the range of what that gene might be able to do. And so as we talked about earlier, we can see signs of strong selection for some domains. Protein kinase domain. Protein kinase is really a critical, important enzymatic activity that is the part of many cellular signaling cascades and domains. And so we can see that there are, more than 250 genes, almost 300 genes that contain a protein kinase domain. So it's been a very successful and selected for domain. Whereas others, we might have, only a small handful. In reverse transcriptase, looks like C. elegans only has 1 or 2 genes that have that domain. And, again, corresponding ranges of these. So it's not just that, genomes are going to duplicate and domains are going to duplicate. We get, equal amounts of roughly equal amounts of domains across all the different types of domains. No, natural selection has a heavy hand in deciding which of those domains are going to be most useful, and a wide diversity provides an advantage, and which ones where having a wide diversity or having extra copies does not really provide an advantage. And so there's probably going to be a much more narrow range of possible functions for these ones that have very few members of the family, and a much wider open range of possible functions for these domains that we find in hundreds and hundreds of genes. And then we also may need to begin to consider how these genes are interacting with other genes, because proteins are not little agents unto themselves. Proteins interact with many, many things. One of the most important things are protein-protein interactions, right? In signaling cascades that bind together to make a usable quaternary structure that need to bind together to perform something like DNA replication. If you look at the process of DNA replication, there are many, many different proteins that all bind together to form that actual functional replication subunit, okay? And so if we look at a network of genes that are interacting, maybe we have 3 genes that are all able to interact with a target, but then we have another one, now we have more complex interactions. And so we can take out components and sometimes add components and do this in a very, very dynamic and flexible way. And so one of the papers that we read points this out, that correlations of mutations have already been found between residues of different proteins and compensating mutational changes at the interaction interface. Meaning that if you get a mutation in one gene, and it makes it maybe even a little bit better at what it does, but that has an interaction point with another gene, there is now selection pressure for that other gene so that when it mutates, it's going to have more flexibility and kind of be able to be dialed in to its optimal interaction with its now changed interaction partner. And so a compensating mutation is a change at a different site, maybe even in a different gene, that makes up for a change that has occurred at another place. And these can be governed by both random chance, but more often are governed by kind of a stepwise gain in beneficial traits, where the mutation occurs originally and overall it's beneficial, although we maybe get a slight detriment because the interaction is no longer as good as it once was. And then later that compensatory mutation comes up, and it allows for that interaction to be as efficient. And so now we have gained both the new mutations function and our interaction is as efficient as it was prior to that mutation. And so these are complex interactions that we need to consider on many, many, many different levels. In the next discussion, I'm going to look at the Bagowski and the Jakubek paper. We'll just do an overview. We'll reinforce some of the things that we talked about here, but there are also some other things that they go over that are worth pointing out.